Hello and welcome to Invertebrate Paleontology and Paleobotany. This is Lecture 5, where we try to answer the question, how do you name a new fossil species? So the first thing we need to define is, what is a species? A species is a population of individuals that are interbreeding with each other, actually or potentially interbreeding, so there's genetic flow in the population, and the population is reproductively isolated from other groups. So the genetics of that group is sort of isolated, so they're reproductively isolated. This might be geographic isolation, or it might just be behavioral isolation. So they don't mate outside of their population. They're under natural conditions. So these organisms are in their natural conditions, and that's how they behave. If you were to collect them and sort of force them to mate, then that would sort of violate the idea of a species. So this concept of a species is called the biological species concept, and it was developed by Ernst Mayr in the 1940s. And his definition is this: a species is an array of populations which are actually or potentially interbreeding with each other and are reproductively isolated from other such arrays under natural conditions. And so most of the time when we think about species, this is the definition that we're using. And it has a lot of caveats to it. So it's basically looking at how the gene flow, the interbreeding of that group of individuals, behaves over a large geographic area. So a good example of species and how speciation occurs, and one of the classic textbooks examples is Darwin's finches, which are on the Galapagos Islands. So when Darwin was on the Beagle, traveling around the world, they stopped at the Galapagos Islands, and while he was there, he was collecting birds. So at each island they stopped at, he would go out and collect bird specimens and carefully label what island these birds came from. When he got back to England, he turned over the collection to an ornithologist who studied birds. The ornithologist suddenly realized that all of these birds were sort of related to a very common bird that was found in South America, but they all were each different in different ways. So some had wide beaks, some had narrow beaks, some were darker colored, some were lighter colored, and that each of those differences they saw in the birds corresponded to each of the islands that they lived in. So what this meant is that each of those populations of birds were isolated reproductively from other islands. So they couldn't fly between the islands, and so they weren't interbreeding, and they became separate species, even though originally they were all part of one species. So this goes back to our idea of speciation, how speciation occurs, where you have a broad distribution of a population that's exposed all over a very large geographic area, and somehow they get isolated enough that gene flow is impeded, and eventually they become a new species. And so we have allopatric, parapatric, and parapatric types of speciation events that happen that deal with that genetic isolation of those populations. Now, biologists are very lucky because they have enormous amounts of data that they can use. Because one of the things they're trying to do is trying to determine whether these populations are interbreeding. And one way to do that is actually take a look at the genetic code of each of those individuals. It's sort of like a paternity test that you can do with each individual of a population to figure out who's interbreeding with who and who's related to who. And so when you're naming new species, this is great because you can actually sample the DNA, the molecular makeup of each of these organisms, and compare them at a population level, so lots of individuals, and see how the population differs in terms of its genetics and how much interbreeding is going on in the population. The genes and the DNA that underlie each organism, the expression of that is called a phenotype. So the expression of the genetic code in terms of form and structure and physiology and even behavior is what we refer to as a phenotype. So in paleontology, we just have the phenotype. We just have that expression of the genetic code in terms of the form and structure that we see in the fossils. So we don't necessarily can see the actual genetic code in the cells, but we can see the expression of that genetic code as exhibited in the morphology and the shape of the fossils that we're looking at. So we do get a little bit of insight into it, but we have to deal with the phenotype, and we don't necessarily deal with the actual genetic code. So one of the things that we see in populations is variation. In fact, you need variation. There needs to be genetic variation in populations for evolution to work. If you had introduced natural selection can select for certain individuals in the population, either for or against certain types of expressions of those phenotypes. And this is how evolution occurs, is through the process of natural selection. And you might have heard of this guy named Charles Darwin, who came up with the idea of natural selection when he wrote The Origin of Species back in the 1850s. Now, since Darwin, there's been some other ideas in terms of how you can actually change and have evolution without natural selection, and that is through what's called genetic drift, or sometimes referred to as neutral theory. This was brought about by a scientist named Theodore Dobansky, who was a Russian scientist who came to the United States, and he was fascinated with fruit flies. He was interested in fruit flies because they only live a few days, and so you can actually study evolution because you can make the flies reproduce with each other. They only live a few days, and you can look at how a population of fruit flies live over many generations, because you get a couple generations every year, actually quite a few. So he did a number of experiments where he introduced populations with different types of frequencies of those phenotypes. So then he would see what happens over time. And he came to a conclusion that what ends up happening is that in the populations that are very small, there's less variation, and so it tends to drive it into extinction. So the variation disappears into the sample. This is also referred to as the founder effect. So if you take a big population of individuals and you only choose a handful of them, you lose out on all the other genetic variation within the population, and that new sampling, that new founding population that's reduced in size has a lower amount of variation of the phenotypes. So if we look at this diagram that has capital A's and lowercase a's, so in the general big, large population, it's 50-50, so even, completely even between the two number-wise. However, if you just were to sample these at random and get smaller samples, so and get, for example, the smallest sample, we have no big A's, it's all little a's, or in the second sample, which is a little bit bigger, we have 6 individuals, we have 4 large A's and only 2 lowercase a's, so there's a different proportion of those A's. So the smaller the population, the more you could drive these changes, and you can do this without necessarily natural selection occurring. So one of the things genetic drift seems to support is that parapatric and parapatric speciation events are probably maybe more common because those smaller populations are subjected to more change. So it can get kind of confusing when you're talking about species. So here we have an example of how you can have 2 different species actually interbreed artificially and produce viable offspring. And the classic example is horses and donkeys. So horses and donkeys, you can breed them and they produce a hybrid, and the hybrid is infertile, so that's the mule. So mule farms and stuff are very useful because they have a better disposition, they're hard workers, but they're infertile. When we talk about plants, we'll talk about crazy things that plants do in hybridization, that is 2 species hybridizing with each other, and dealing with some of the chromosome splits that are kind of crazy. So it can get really confusing of where you delineate between one species and another species. For example, if mules were fertile and could produce viable offspring, it would seem to suggest then that horses and donkeys are the same species. But the fact that they're infertile, and in fact that only this mating process occurs because we humans desire it, that's what produces those hybrids. But it can get kind of confusing too when we're trying to deal with delimiting different types of species by using the biological species concept. So what a lot of biologists do nowadays is they go out into a population that they're studying and they sample lots of blood or tissue. they run DNA sequencing on that tissue, and then they run it through big, huge analyses, and they look at how closely similar those genetic codes are, and sometimes they can recognize a group or a population in their sample that's genetically different from all the others, and they can argue that maybe this population is isolated from the other populations and not interbreeding with the other populations and represents a new species. So this represents what's called the modern species concept, which has a not very eloquent, not very good definition. A species is a species if a competent specialist says it is. Because we're looking at all of this code, we're looking at how distances and genetic makeup and whether one population is different enough and isolated enough to represent a different species or not. So the key thing is trying to determine if the two populations are interbreeding or whether they're isolated and not interbreeding with each other. So it can get kind of tricky. So now let's talk about the morphological species concept. This is the concept that's used in paleontology because we don't have genetic code of each of the individuals that we're looking at. They're just fossils. We can then look simply at the morphology, at the shape. So we ask the question, do they look different or not? And in paleontology, it's fairly simple to determine this. We can run some analysis and stuff, but the key term is, do they look different enough? So if you took two fossils and you handed them to somebody, you can say, do these look the same or different? And someone can say, yeah, they're different. But the real key is basically in our ability to look at the differences in the morphology and whether we can actually distinguish each of them from each other. Sometimes, of course, it can be tricky and you have to use statistics to figure it out, but sometimes it's fairly straightforward and fairly simple. There's a certain shape, there's a certain extra spine on the fossil that we can look at, and so that's how we designate differences in the fossil record. But we got to be careful, right? We got to be careful that we're not dealing with different aged individuals, we're not dealing with aberrant specimens or pathologies, that we're not dealing with all these other things that we've talked about this semester. So morphological species, do they look different? And we're hoping to have a big population that we can compare between the two. Now there is a problem with this in paleontology, and one of those problems is that species change over time. And so in paleontology, we're dealing with huge amounts of geological time that we're looking at things. And of course, species do change in the fossil record. So the real problem is then, where do you delimitate the temporal boundaries of a species? G.G. Simpson was a famous paleontologist at the American Museum, and he introduced a new concept, and that is the evolutionary species concept. So the evolutionary species concept states that a species is a lineage evolving separately from others and with its own unitary evolutionary role and tendencies. So let's look at how Simpson would sort of classify species in the fossil record. So imagine that you have a species that's changing over time, changing linearly over time. This could be getting larger, getting a big horn, growing an extra foot, but there's some sort of trend over time, and we have intermediates between the two. But, however, the fossil record's not perfect, and we have gaps that are between each of these little normal distributions that we see on the graph. So what a morphological species concept would do would say, hey, we got species A down at time A, we got species B at time B, and at species C at time C. So we have three different species, and they're separated by these temporal gaps, where we have gaps in the fossil record. Simpson, with the evolutionary species concept, would say, no. This is one lineage, this is one species, we'll call it all species A. So that's the evolutionary species concept, and it's used quite a bit to sort of group things together, but it can be kind of problematic because you're grouping a whole lineage all together, and there might be some differences in there that you're not seeing. The interesting thing is that with the increasing number of fossil specimens that are being studied, that long evolutionary species that show gradual trends like this are fairly rare, and we're going to talk a whole lecture on punctual equilibrium and discuss this in more detail of why that might be, that these general sort of changes in the fossil record are kind of rare. So another way to do it is basically naming the species when they split. So this is a diagram that Niles Eldridge put together, and it's a wonderful diagram because it illustrates how the isolation, the genetic isolation, is the key point in distinguishing two different new species. So in this case, we have a species A, population. There's a geographic isolation event, and one species, species C, becomes kind of flat and squat, and species C becomes flat and squat, and species D becomes kind of long and pointy there. And so then we can distinguish them based on morphology, but when we get back to the very beginning, at that root there, it becomes hard to tell, and so they recognize a separate species. So here we have three species. We have the species before the split, and then the species after the split, and they can be distinguished by different morphologies, although there's a continuum there. And so a lot of people will define a species when in the sample that they're looking at you see this shift or change that happens in the fossil record, kind of an abrupt change. But it's very rare that you actually get a very gradual change, and the reason for that is that a lot of the evolution is happening in these peripheral populations, small population sizes, where we see the most amount of change. All right, so now let's talk a little bit about the process of going through and naming a new species. So imagine that you find a new fossil species, you're really excited, you've gone to all the museums in the country, you've pulled out drawer after drawer after drawer, and you cannot find anything that's like it. It looks completely new, you're very excited, it represents a new species. So the first thing you want to do is to specify your type specimen. This is referred to as the holotype. The holotype is going to be the specimen that everybody's going to come to look at to determine if they have a new species and to distinguish that species. So you need to designate a holotype, a new specimen. You may also have a number of other specimens, maybe from the same locality, and you can group those with the holotype into what's called a hopodigam. Now, before you begin, you got to read through the rules, because there's some rules in naming species that you got to go through. If you're naming an animal, you got to go through the International Code of Zoological Nomenclature, the ICZN. And if you're naming a plant, a new plant, you got to go through the International Rules of Botanical Nomenclature. And both of these have websites, and I'll have links on our class webpage that you can check out each of these rules. So I want to go through a couple of the rules, some of the more important ones, but definitely if you're naming a new species or thinking about naming a new species, definitely check out the codes. So the first the first rule of naming a new species is you must publish in a qualifying medium. That means you must publish in some sort of academic journal or publication that has a fairly wide readership, and that way people can access your description of this new species. It used to be, not that long ago, where you couldn't publish a new species in a website or on the internet. It had to be a printed journal. They've changed this, and so now you can publish in a journal that's only online or available electronically, but you have to register it through some sort of database. And the one that most electronic journals have you register it is Zootaxa, although I think it might change to Encyclopedia of Life, which is a group of very large consortium of web pages. Each web page is dedicated to a new species. It's a huge index of all the species, trying to get all the species that ever existed into it, and it's maintained by the Smithsonian Institute, and its URL is eol.org. You should check it out. So you got to have a qualifying medium that meets the criteria. And the next thing is you got to come up with the name, and the name is a binomial name. So the first part of the name is the genus, and the second part is the species, and together they must be unique. So, for example, humans is Homo sapiens. The genus is capitalized, and the species in lowercase, and it's italicized or underlined in all scientific publications. And this was the method that was developed by Carl Linnaeus back in the 1600s. He came up with this idea of using a binomial. So if you're naming something new, a new species, you got to come up with the species name and then decide which genus, or even create a new genus to put it in. So you have a binomial name. Now you can't name a species after yourself. Although colleagues can name a species after you, you can't name a species after yourself if you're an author on the paper. So sorry, can't name a species after yourself. Species don't necessarily need to be Greek or Latin. In the olden days, everything was Greek and Latin in science. They can now be in any language. You can even have unpronounceable species names. People have named species after rock stars, after their favorite Harry Potter characters, after Lord of the Ring characters. You could come up with anything. Anymore, they're getting more and more unpronounceable. So you can name them kind of anything and put it out there. Even curse words is acceptable, though it's really frowned upon. The type specimen, that holotype that you designate, has to be housed at a public museum so it can be examined by other researchers. This can be a problem in paleontology where maybe the specimen is a new dinosaur or a new really cool fossil and it's in a private collection or is collected by an amateur who doesn't want to donate it to a museum. They want to hang on to it. In that case, it can't be a new species because no one can kind of knock on their door and say, hey, can I look at that specimen that you described as a new species? So they have to donate it to a museum and it has to be publicly accessible. That means people can come to the museum and actually pull out drawers and take a look at that specimen and compare it so they can decide if they have a new species or not and actually come and study the specimens. So it needs to be housed in a public museum. Species names can't be named anonymously. You have to have your name and the date, and that's really important, and I'll talk a little bit about that later. So you can't just name it with anonymous name. One of the key things about naming species is the law of priority. That means that whoever names the species first gets the credit. So if somebody beats you by a day or 2 days or a few hours to get that into press, that becomes the publication. You need to be careful with this because sometimes what can happen is, say you're naming a new species, you're really excited about it, and you then contact, say, journalist, and say, we're naming a new species and we're going to name it, Gubernaculum cuckoo. And they say, that's a great name. And they run a story and they publish that story before you have a chance to name it. You can run into some problems. And that's happened in the past, and some people kind of don't worry about that happening too much. But you want to be careful. You don't want to reveal the species name until that publication comes out and is actually published. Now let's talk a little bit about going through how you would systematically name your new species, write up a description, put it all together. So the first thing you want to do is you come up with your species name, your new scientific name that you're going to call it. And you're going to write it to a journal and try to get a manuscript published in one of these journals. For paleontology, the Journal of Paleontology, which oftentimes publishes new species, but there's many, many other journals, scientific journals that are out there that publish species descriptions. So you have the name, you're going to have the taxonomic references and any synonyms that you're going to put in there. You want to include your holotype, your type specimen, and you want to include information about where it was found and its present repository, what museum you put it in, and oftentimes you'll put the catalog number and the locality number of the specimen so people can actually come to the museum and take a look at it. You then want to write up the diagnosis, how you distinguish this species from other species that are out there. You're going to write up a very detailed description. You're going to include photographs, great photographs in your description. You're going to include all your measurements and other numerical data. You're going to have a discussion, what's the importance of this new species, how does it relate to other species. You're going to include the range, the geographic and stratigraphic information about the species. You're going to talk about the habitat and the horizon that the species is found in. And then you're going to list the material examined. This material could be other specimens of different species that you examined and looked at. So you can have a long list of all those specimens that you looked at and compared it with. You then want to put your cladistic analysis, and the cladistic analysis is going to show the relationship between the new species and previously named species. We'll talk more about cladistics and phylogenetic analysis in a separate lecture. So you put all that stuff in. That's a lot of work to name a new species. You have to put all this stuff in and get this paper published. So let's talk a little bit about the different types of type specimens that you might encounter in the literature when you're kind of reading through a lot of these descriptions of new species. So let's talk about type specimens. So the first one is the holotype. That is the one that you mentioned in the paper. You designate it as the holotype. It is the official specimen to look at when you want to compare your specimens with other specimens. It's the one to go to. It's the gold bar, the gold standard. Sometimes you have other specimens that you want to include that might have a unique characteristic that's not exhibited in the holotype. Those are called syntypes, and you can add a syntype that may be a little bit better. Sometimes syntypes can be added to previously described specimens where they have a better specimen that they want to introduce as a syntype. The other thing that happens a lot in invertebrate paleontology is you find lots of specimens associated with it, and you want to designate lots of paratypes. These are specimens that were found with the holotype or closely associated with the holotype that may show different features. For example, in fossil mammals, you get teeth that preserve a lot, so you may have a new species based on upper dentition, but you want a paratype that's a lower dentition that we can compare it to. Another type of type that you can have is a lectotype. A lectotype is where you have an issue where the holotype is not very good, and you want to designate one of those paratypes or syntypes as the new holotype, and so you put that into, you make it that lectotype, and you get rid of the holotype. But you don't call it a new holotype, you have to call it a lectotype, because the holotype stays the holotype forever. Sometimes you have a specimen, you have your holotype, and the holotype is lost. The museum misplaced it, the museum got bombed, there's a war, and the holotype's gone. And so instead of creating a new holotype, you create a neotype, and a neotype's a new specimen that you say, this will replace the old holotype, but we're going to call it a neotype, because, who knows, maybe the holotype will be discovered and we'll have a specimen of it. Sometimes in older publications, the scientists that originally described the species may not have designated a holotype. And this happened a lot in the 1600s and 1700s when they still were kind of just naming everything. So they didn't designate a particular specimen to be the holotype. In that case, we can designate a plesiotype, and that is that we find a good example of that species and we designate that as a plesiotype. Sometimes you have lots of specimens that are collected at the type locality, the same locality that you found that specimen in, and that is called a topotype. And those are also oftentimes included in collections. So those are all the different types of types that you can have in museum collections. So those are some terms to be aware of when you're digging around in museum collections and finding paratypes and topotypes and neotypes, what those things are when you see that listed in a catalog. All right, so let's talk about circumstances that you might encounter where you have to come up with a new species name because of some sort of something wrong in the books. These are basically times where you have identical names for two different species. There's what's referred to as primary homonyms, and this is where you have a species. Usually these are genera because genera have to be unique names. So here's a good example. So there was a fossil brachiopod called Margarita. It was named. Some researchers later on found a fossil primate, and they thought, Margarita's a great name. We've been drinking lots of margaritas. We should name it Margarita. So they named it Margarita. Then someone said, wait a second. Someone's already named a brachiopod Margarita, so we can't name the fossil primate Margarita. So they just changed the spelling of Margarita with an H, and that way then it's okay. So you have Margarita spelled with an H is the primate, Margarita spelled with the R is the brachiopod. Sometimes you can have what are called secondary homonyms, and that is where you have circumstances because of the binomial names where you get the same. So in this case we have Margarita stevensi, that's the species name, and we have Parasagius stevensi. But a researcher discovers that Parasagius stevensi should be included in Margarita, the genus Margarita. So now you're going to have Margarita stevensi and Margarita stevensi, and they both have the same name. So you have to come up with a new species name, maybe it's Jeffersonia, for that that used to be Parasagius stevensi. This actually kind of happens more frequently than you think, because oftentimes there's a famous researcher who everybody loves to name their species after, and they work on one group, and so there's a bunch of Stevensi because she just named all these species, and so they all kind of get grouped together. So it can happen that way. So then you have to come up with a new species. Sometimes you need to get rid of species names. For example, if you have synonyms, and this is what happens when you have two different names applied to the same species. So for example, you got a whole population you've been looking at, and one researcher thought one should have been a different species, another one should have thought there was a different species, and everybody's disagreeing. You come in and they're like, hey guys, this is all just one species. You can group them all together, and that is a synonym. A good example of that that's debated quite a bit is the debate between Eohippus and Heracitherium, whether Eohippus should be the designated name for New World early little horses and Heracitherium for the Old World early horses, or if they're the same and should be together. And there's a lot of debate on that, and I won't go into it, but that's kind of an example of why you might have a synonym. The other one that's very common in paleontology is nomium dubium. So nomium dubium basically means dubious names. And this happens a lot in paleontology. So a researcher goes out, they find a really fragmentary fossil, but they think, oh, it's a new species, I'm going to name it a new species. And they name it a new species, but it's only a fragment of something. And then researchers later find incredible fossils, and they realize that that fragment is just undiagnostic. There's no way you can tell if it's species A or B or C or whatever. And so they make it a non-dubium, which means a dubious name. So that's when the type material, the holotype, is not good enough to diagnose a species. And that happens quite a bit in paleontology, because the more fossils we discover, the more we start to understand that there are certain features that we can use to diagnose those fossils. And if those are not preserved in those early holotypes that were designated by researchers 100 years ago when they came up with the name, then we just make that non-dubium and throw that name out. And it happens quite a bit in paleontology. All right, so thanks for watching this lecture, and hopefully this will give you some ideas on if you ever discover a new fossil species and are interested in naming it. If you're watching this video from the internet and are interested in taking a course in paleontology or geology at Utah State University, check out the department website at geology.usu.edu. And if you're interested in the research and who I am, check out my website at benjamin-berger.org. Thanks for watching.